Substitution, as it sounds like, is when something is substituted for another. Deletion is what it sounds like. It's when a nucleotide, well, let's start with substitution again and use the word nucleotide. When substitution occurs, a nucleotide is substituted for a different nucleotide, which could cause a change, right? That's a mutation. Deletion is when a nucleotide disappears or is deleted from the sequence, which would cause a difference in your DNA, which would cause a mutation. And then insertion is when we put a nucleotide in that wasn't there to begin with. So that would cause yet another change or mutation. Okay? So let's look at our 3 different types. First of all, substitution. Okay? So here we have our order of nucleotides. Let's say we have green, do another green here and a green here. We have red, 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 and red. We've got our green and our green. Okay. Then we have brown, 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 and blue, 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 blue. Okay? This is our gene. Okay? This is our sequence. This calls for, let's say, round fingertips. Okay? Now, the first one that we have that can go wrong, the first mutation, is substitution. What happens is, as the DNA replicates, when it makes a new copy, the copy that it makes, let's say it substitutes, instead of blue here and brown, it's going to substitute pink in there, right? And then it's going to make that green there. Okay? So that's just changed one little piece of our genetic information, right? Right here is where the substitution occurred. And maybe that doesn't cause any difference in your gene because it's just one little piece. But maybe it does. Maybe it caused you to now have flat fingertips. Okay? And bam. You're like, oh, why are my fingertips changing? What's going on? Mutating. Becoming totally different. Well, what's happening is all the way down at the molecular level, inside of your cells, your DNA changed just a little bit, and it caused your fingertips to slowly become more flat, right? As your cells reproduce. Now, the next one is a little more involved, okay? This one is called a deletion. So we have our DNA again, and I'm just going to do 4 this time because it's quicker. Okay, so we've got pink, pink, and then green, green, and blue, blue, brown, brown. Okay, so as the DNA is replicating, what happens is this one is just taken out, right? Now the order is totally different because everything after that deletion, let's say it was all of these nucleotide base pairs that happen after that, they get moved up. They get moved up in that order, and now our protein production is going to be off, right? The same thing can happen with insertion. So we'll call this deletion, sorry. Deletion. Bam. Now, what about insertion? Insertion is just like deletion, except the opposite, Uno reverse, right? We've got our DNA sequence. It started off with blue, blue, blue. Let's go brown, 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 and green, 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 and green, and pink, 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 right? Okay, so there we have the same thing. Now what happens when DNA is replicated? What do? We stick in another nucleotide pair. It gets inserted into the DNA. And again, all of these nucleotide pairs after this here, all of those nucleotide pairs gets moved backwards in order. Okay? And protein production gets messed up again. Now, how does protein production play a part in this process, right? Why does this change our protein production? Why does deletion change our protein production? Why does substitution change our protein production? Let me show you why. All right, so now we have our process of transcription and translation is when, we make a copy of, or a single-sided copy of our DNA that goes out of the cell into the ribosome to make proteins. So here we have our piece of protein going to the ribosome, ribo, right? And it has our order of nucleotides that tells us what proteins to make. And as it goes into the cell, it decides what proteins are going to be made, right? And here you go. My gosh, Mr. Neale, just get on with it, right? Okay? So we have our order of nucleotides going into our ribosome, which tells us which proteins are going to be made. Okay? But for every 3 nucleotides, every 3 nucleotide pairs, or sorry, not nucleotide pairs, but for every 3 nucleotides, an amino acid is coded for. Okay? All of these code for different amino acids. Every 3 nucleotides in a row codes for an amino acid. And as we combine amino acids, we create proteins. Well, let's say this is amino acid number 7. This is number 7 also. Let's call, well, no, that's different. So we'll call this 9. Let's see, is this the same? No, we'll call this 11. We'll call this one 1. We'll call this one 3. This one's amino acid number 20. And this one's amino acid number 8. Okay? So we have 7, 9, 11, 1, 3, 28. What does that mean, right? That means as we're combining these amino acids in that order, we're going to create the protein that tells our hair to be curly. Right? This sequence of nucleotides caused this sequence of amino acids to be put together, which caused the protein for curly hair. Ta-da! Right? And as we notice, when DNA replicates, and if we have a, let's say, a deletion, let's say this one just totally disappeared. off of our DNA, then we have an order that goes 7, 9, 11, 1, 20, and 8. That changed my amino acids, which changed my protein, and now this is the protein for nothing. It's just a worthless protein, and your hair doesn't get told to be curly, so your hair kind of gets funked out, right? Your hair is like, oh, I'm just going to be whatever. Some hairs will be curly, some hairs will be straight. It's just going to be weird, okay? Or let's say we had insertion. So we had another nucleotide come in, or sorry, more nucleotides come in, which coded for another amino acid, and let's say this 3 is back, but then we inserted a 14 here. And now we have yet another combination of amino acids that's going to cause a new protein, and this protein, say, causes straight hair. And that insertion now caused a new protein to be made, right? If we have substitution and that becomes green, then this protein or amino acid becomes number, let's call it number 19, and yet another protein is developed. And then let's say we have a whole nother movement of nucleotides, insertion or deletion, and now this circle doesn't circle those 3 nucleotide combinations, right? These 3 that code for an amino acid. Let's say everything moved up or moved over because of deletion or insertion. Now we're going to circle, let's say, these 3 are now a new amino acid. We have these 3 are an amino acid, are coded for a different amino acid in this one. And now we've changed our numbers yet again. Let's call this 2, 4, 6, right? So now yet again we have another protein that's being coded for instead of what the original DNA called for due to mutation, which is either substitution, deletion, or insertion. And substitution, deletion, insertion causes the nucleotide order to change, which causes new proteins or different proteins to be made inside the ribosomes, which makes your traits change. All right? Hopefully that got through to you. If you are still confused, watch the other videos, read the material. If it's all just flying over your head and you're still confused, email me. Let me know what your questions are. Your fellow students might be helpful with you. And then when you're doing your projects or your your assignment, which is to make pictures or models or a video, please make sure that you're including how DNA replicates, what mutations occur, the 3 different mutations and what happens, and then how they affect protein production. Okay? So that's a lot, I know, but you guys can do it. You've got plenty of time, right? That's all we've got. So keep on keeping on, and we're doing this, guys. I miss you. Can't wait to be back in the classroom, and go get them, Tigers.